Hi, welcome to lecture on environmental adaptation for animals. Now we will continue with a few examples on the hormonal regulation of metabolism and homeostasis. So something that is important for all kinds of vertebrates. Let's start with the metabolism because that's the simplest way. So we have learned from the school books that, okay, after the meal, the glucose concentration in the circulation is high, but then you add some insulin into circulation and that will reduce the glucose back to normal level. But quite often we are eating also other nutrients than just glucose or sugars. And that's why the control of this metabolism and catabolism is we have more regulators than just the insulin. And quite often these regulators are interacting. We already learned in the previous videos that, okay, we have this co-regulation. So the insulin is less effective to reduce the glucose levels if there are some other factors present in the circulation. So let's have a short view of this. So the glucose concentration in the blood can be reduced by having insulin. But it also can be increased by having another hormone, glucagon. And we already learned in the previous videos that glucagon and epinephrine are very effective together to limit the glucose uptake, what the insulin is triggering. But also glucocorticoids and the growth hormone are doing the same. So we have 4 hormones that are limiting the effectivity of insulin. And this glucocorticoid then is increased by thyroid hormone. So now we have 5 factors that are affecting on the insulin sensitivity for this process. But also the insulin is controlled. We have osteocalcin that is increasing the insulin secretion, and we have osteocalcin is coming from the bones, and that affects also in the fat cells that are releasing adiponectin that is enhancing the insulin sensitivity of the receptors. So we have actually now quite large number of different hormones that are interacting and regulating this glucose concentration in the circulation. So we have insulin that is stimulated by high glucose. Then we have glucagon, epinephrine, glucocorticoids that are either with low glucose concentration or this sympathetic stimulation and or stress. And then we have other hormones like thyroid hormone, growth hormone, endocrines, and osteocalcin that are regulating this system. And they can be continuously secreted or mediated by growth hormone, releasing hormones, or etc. And they have different stimulus on these sugar levels in the circulation. So the insulin is increasing this uptake in the skeletal muscles and the fat cells, and formation of glycogen and triglyceride synthesis. So it's turning on the glucose metabolism and turning off the lipid catabolism. So we are using more glucose as the energy source. But then we have glucagon, epinephrine, and glucocorticoids that are making it in the other way. So turning on the lipid catabolism and inhibiting the insulin release. And then we have thyroid hormone and growth hormone, androgens, and osteocalcin that are also affecting on this turning on lipid or glucose catabolism. So using either sugars or fat as the energy source, either directly or indirectly by increasing this insulin mediated uptake. So what happens if we have different kind of diets? First, this school book example of the sugar diet. So we have high glucose concentration, and then we have high insulin concentration, and at the same time the glucagon release is inhibited. So the insulin is secreted from pancreas beta cells, and at the same time the release of glucagon from the pancreas alpha cells is inhibited. And how this insulin is affecting, it's increasing the activity of glucose transporter GLUT4 in the skeletal muscles. But it's not affecting on the brains or liver, because in those cells the glucose uptake happens all the time. And it can be boosted with osteocalcin or adiponectin. But then, when we have a protein diet, you can see that after a protein meal, the glucose concentration is increased, but the glucagon release is boosted a lot. So that's why during the high protein meal, the glucagon is released, and this reduces the glucose uptake. We have already learned in the previous videos that if you have both these hormones, so the glucose uptake is slower, and then they are turning the amino acid and glycerol to glucose in this gluconeogenesis. And they are also stimulating these adipose tissues to break down triglycerides. So this means that during this protein diet we are actually breaking quite a lot of fat, and we're also making more sugars from our other energy sources. So it's not just the between the meals hormone and the protein containing meal trigger the release of both hormones in the circulation. And this then affects that if you are having stress like exercises, and that's affecting on the epinephrine concentration, and then you are having protein diet, etc. So it's means that the insulin is quite ineffective. So you might have quite high concentration of glucose if you are having sugar in your raised sugar levels in the circulation. But then the other way of example is on the salt and water balance, and of course that's coming on the nephrons and the kidneys. We have antidiuretic hormone, meaning vasopressin, and it's exists in all vertebrates to regulate the water uptake in the kidneys. And these kidneys are operating with a filter and reabsorption mechanism. So they filter all kind of small molecules from the circulation and then uptake water and the useful particles back. This antidiuretic hormone is affecting on the epithelial membrane of the collective duct over here in this picture on the left one. So it's affecting on the amount of water channels on the epithelial membrane. And if you have water channels, it makes this apical membrane permeable for water. Let's have a closer look on that later. But then we have also the salt balance. We have renin, angiotensin, aldosterone system that is regulating not water channels, but now on sodium pumps and ion channel expression. So when we have this antidiuretic hormone over here, it's on vasopressin, it's affected on the receptors. This receptor is G protein mediated, so there will be cyclic AMP and protein kinase. And the protein kinase is then activating that these vesicles containing these aquaporins will fuse with the apical membrane. and that's why you get more water channels on the apical membrane, allowing water to flow from the lumen to inside this apical epithelial cell. And because there is a completely different concentration of different substance also in this on the other side of the epithelial cell, then this water is moving to this peritubar capillary. So we can have, by just adding these water channels, we can passively pump back the water from the lumen to back to the circulation. But what about then the salt? We have the nephron, and on one part of the nephron we have this brown capsule, and beside that we have two places for hormone releases. We have macula densa that is releasing a paracrine. So now it's not a hormone that is all around in the circulation, but it's also only affecting on the neighboring cells, and they are just there beside them on the these juxtaglomerular cells that are secreting renin in the low blood pressure. And what happens then? Okay, we have angiotensin again that is produced constantly in the liver, and if we have the renin, because there's a low blood pressure, this angiotensin again is modulated to angiotensin, which is modulated future to another kind of angiotensin, and then starting the aldosterone secretion. And the aldosterone is affecting on these on the distal tubules and early connection ducts to increase the sodium reabsorption and potassium secretion. How you can do that? By simply sodium potassium ATPase. So you are pumping sodium out from the cell. You are pumping it inside the cell, and then you are pumping potassium out. And that's why you can then reabsorb the sodium. But at the same time, this angiotensin is also stimulating the construction of systemic arterioles and promotes thirst and stimulates the vasopressin secretion. So we have extra volume fluid is increased, and that will increase the blood pressure. But it also affects on different steps. So it affects on the urinary bladder, sweat glands, salt glands, salivary glands, intestine, in different kind of organisms, and always how it works, it affects on the sodium channels, potassium channels, sodium potassium ATPase. But what about then how to increase the loss of water and sodium? For that, we have atrial natriogenic peptide. So if the blood volume is high, these atrial muscle fibers are stretched. So there's a little bit too much blood in the circulation. And this will cause this atrial natriogenic peptide release, and one part of that, it's inhibiting this antidiuretic hormone renin and aldosterone release. But that's not enough. It's also increasing the glomerular filtration. So more blood is going in the kidneys to be filtered. And at the same time, it's inhibiting the reabsorption of sodium and increasing its excretion. So now you are limiting the amount of sodium channels, or limiting the activity of these sodium channels, or limiting the sodium active pumping, this reabsorption. And these are together affecting the, okay, now there's more sodium lost, more water is excreted, and that causes that, okay, the blood volume and pressure will be downregulated. Okay, the third example is on the calcium mechanism, calcium metabolism, sorry. And it's important because it's essential secondary messenger in all kinds of cells. And in several cells, the regulation is actually based on the entry of extracellular calcium. For example, in the synapse. For example, in the heart muscle, or in the smooth muscle, etc. So they can't just rely on the intracellular calcium stores. And it's regulated with 3 hormones. There is a parathyroid hormone, PTH, causing that, okay, more calcium and less phosphate in the circulation, and it stimulates the vitamin D activation. Then we have active vitamin D that is acting as a booster. So there's more calcium again. And then we have calcitonin that is causing less, so downregulating the calcium concentration in the circulation, at least in some animals. So what happens when we have low calcium? So we have chief cells in the parathyroid gland. They have a sensor for extracellular calcium. And the calcium is binding on the receptor and mediating by G protein mediated signal, and which means that, okay, now when there's low calcium, this system will trigger the parathyroid hormone release. And this parathyroid hormone is affecting in the target cells why G protein and cyclic AMP mediated signaling pathways, and enhance this calcium release or calcium uptake by several mechanisms. And this is boosted with this active metabolite of vitamin D. So we have parathyroid hormone that is affecting on the bones. We have their osteoblast and major osteoclast. So this means that, okay, now more calcium is released from the bones to regulate the calcium concentrations back to normal. But that's not enough. So it's affecting also in the kidneys to increase the calcium reabsorption. And it's also affecting modulating the vitamin D that is normally in the skin. Then it's turned in the liver and in the kidneys for this active form. And then these are together affecting on the get higher extracellular calcium. And as you can see, this vitamin D is boosting the signal. So it's enhancing the calcium reabsorption in the kidneys. It's enhancing the effects of parathyroid hormone in the bones. But it's limiting the conversion of this active form of vitamin D, so there is also a negative feedback. But even this is not enough. So this vitamin D is also increasing the intestinal calcium absorption. So from the food we are moving more calcium to the circulation. And of course this is also affecting on the extracellular calcium concentration. And of course sooner or later we are having enough calcium, and this causes negative feedback for these chief cells. But what about then in the high calcium? Well, the mechanism is pretty much the same. The place where it's sensed is pretty much the same. It's not in the parathyroid, but it's now on the thyroid gland. And there is a C cells that are working exactly on the same mechanisms. But now, when we have high calcium, they release calcitonin. And this calcitonin is affecting on the bones. So limiting the calcium release from the bones. But it's also limiting the calcium reabsorption in the kidneys or the conversion of active form of vitamin D. And because there's less this active vitamin D, also the intestinal calcium absorption is limited. And together these are then downregulating the extracellular calcium in the circulation. And this will have the negative of positive feedback on the in the thyroid and parathyroid glands. So that's why we can have two different mechanisms that are working in the opposite direction, and then we can have quite nicely the same amount of calcium in the circulation from day to day. Thank you.